Thank you very much for this kind introduction. And my first slide will take you several hundred years ago to the story of the French Revolution to convey the message that revolutions are often recognized in retrospect. And those revolutionaries shown in this slide, I believe they were not aware of the fact that they are going to change the face of democracy for decades, for hundreds of years. And this is what I believe is going on in the field of clinical oncology. We see revolution of molecular targeted therapy of cancer, a revolution that started about 100 years ago with the idea of Paul Ehrlich, a chemist who worked in Frankfurt on dyes that he noted specifically stain some tissues and ignore neighboring tissues. And he came with a brilliant idea that it would be possible one time to direct drugs to some tissues and ignore other tissues. This is what he called the magic bullet idea. And indeed, we see with this slide the development stepwise of drugs like trastuzumab and rituximab and Gleevec over the last 10 or 12 years. And what enabled this revolution was what I called the Oncogene Decade, 2 decades between the 1970 and the 1990 that allowed us to resolve pathways that convey, relay signals from the outside of the cell, from the world, the extracellular world, all the way through the membrane to the cytoplasm and to the nucleus, to the genome. And amazingly, many of us were reporting oncogenes, new oncogenes. But in retrospect, turned out we were all working on the same pathway, a pathway that leads from the plasma membrane, from growth factors, through receptors like VEGF, like EGF, like FGF, down to effectors like RAS and RAF, and PI3 kinase, a major hub in this pathway, all the way down to proteins, transcription factors like MYC and P53. And why growth factors and the receptors are so important in cancer and in cancer therapy? The reason is because they are controlling all phases of tumor development and metastasis. Namely, when a cancer cell attracts a mutation, growth factors are responsible for the expansion of the clone, the initial expansion of the clone. Later on, they are responsible for angiogenesis. And later on, they are responsible for metastasis, for the motility, penetration, and invasion into blood vessels and lymph vessels, and colonization of distant organs. And perhaps most important, growth factors are responsible to survival under cytotoxic drugs and radiotherapy. They are those who allow tumor cells to evade, to resist those therapies. And that is the reason, I believe, why we often use those cytotoxic drugs in combination with targeted therapies, why the combination is so effective. Because growth factors are important also in this final step in tumor progression. Now, today I would like to take you into signaling complexity, because we believe that signaling complexity underlays some of the observation you clinicians are making, like patient response and patient resistance and secondary resistance. And this is kind of an electronic board of the EGF receptor signaling pathway prepared by Sony Corporation in the System Biology Institute in Tokyo. And what I would like to tell you is signaling is extremely complex. Nevertheless, resolving the complexity is so important because the reward, like I will exemplify today for EGF receptor and HER2, the reward is extremely large. For example, EGF receptor and HER2 are targets for so many drugs, maybe unprecedented case in oncology. I list some of them: gefitinib, erlotinib, lapatinib, cetuximab, panitumumab, trastuzumab, and more are coming. Also interesting to note that those drugs and EGF receptor and HER2 are targeted in several different clinical indications. Again, I would like to list some of them: lung cancer, breast cancer, colorectal tumors with cetuximab, head and neck cancer, and pancreatic cancer with erlotinib. So certainly it is important to understand complexity. And what I would like to tell you today, that we are fortunate. We are fortunate to be able to see the former electronic board of the system, those that existed in world on Earth some 800 years ago, for example, in little worms, just 1 millimeter long. So let's consider such a simple electronic board, A interacting with B. Let's assume A is EGF, B is EGF receptor. Now, upon genome duplications that we know occurred in evolution once and again, there is not only one A, there are two A's. And B become 2B, and they continue to interact. And now imagine another genome duplication. A become 4 A's, and B become 4B, and they still interact. This is important. So they generate a two-layer network with many, many interactions. This is the origin of complexity. And when you treat with a drugs that inactivate A, there are so many other components and so many other interactions. And of course, this is simplified. We are dealing with networks with maybe not only 4 A's, but maybe 20 A's, not only 2 layers, but maybe 10 layers, and you get enormous complexity. So this is one way to present the complexity of the network, in which the many growth factors of the EGF family binds to 10 different homo and heterodimers, including heterodimers of HER2. And they signal all the way through the cytoplasm to the genome. Primarily, the input is cell proliferation, but also cell migration. And what I would like to convey to you is what we, in the basic research part of it, would like to consider the biological complexity and networks in terms of engineering. We take lessons from electronic engineers and chemical engineers to resolve why the network developed this way. What is the logic of the network? And one way to understand the logic is robustness, the fail-safe property of the network, how it overcomes perturbations, inhibitors, mutations, drugs, if you want. So part of the robustness is system control, which is shown here. Positive control, these are the ligands, these are the receptors, all the way down to the PI3 kinase pathway, MAP kinase pathway. So system control holds the robustness. And today I would like to tell you about positive and negative feedback control. And as a rule of thumb, positive feedback control is enhanced, like TGF-alpha, HBEGF, ligands of the family VEGF, enhanced in cancer, and negative feedback control is defective. And I would like to show you one example and move on to more examples. This is EGF receptor in green, GFP labeled, stimulated with EGF, which is red. And every time they meet each other, they make a complex which is initially yellow. Now, the complex make a vesicle that undergo endocytosis. Soon after, it degrades the green, EGFR, and remain red. And in a few minutes, you see that there is almost no green EGFR on the cell surface. This is negative feedback regulation, immediate, within a few minutes. No need for new messenger RNA. And this has to do with receptor phosphorylation, followed by ubiquitination, all the way down, sorting to degradation in the lysosome. And this is a negative feedback control, which is defective in cancer. One manipulator of the system is HER2, because when HER2 is overexpressed, it makes heterodimers that switch EGF receptor. Not anymore it goes through the endosome to degradation in the lysosome, but it recycles back and once again to the cell surface. This is a negative feedback control, which is defective. Now, another more complex, more subtle defect has to do with messenger RNAs that are stimulated within seconds or minutes after stimulation with EGF or related growth factors. These are the immediate early genes. Some are upregulated several-fold right after stimulation. For example, C-FORCE, C-MYC, C-JUN. They are all included here. But slightly later, there is a wave of delayed early genes which are there to inactivate, to suppress the oncogenic transcription factors like C-FORCE and C-JUN. And what we found is that those, I show it here. This is a DNA array, time 0. 20-minute stimulation, many oncogenic transcription factors are activated in red and yellow, but slightly later, some delayed early genes, like suppressor of transcription, like phosphatases, MAP kinase phosphatases, like RNA binding proteins, they are upregulated. Now, this switch is defective in cancer. And in a subtle way, this is an example in which we compare malignant and normal tissue and we use DNA and RNA binding proteins, transcription factors that are included in the delayed early gene. And overall, with some exceptions, delayed early genes are down-regulated in cancer. And we looked at 22 tumor types. Turned out over the last year, we started to work on microRNAs. MicroRNAs are tiny molecules, RNA molecules, that bind to messenger RNAs in a specific way and are responsible for their degradation. So they control messenger RNA. What we discovered, to our surprise, is that the microRNAs are also dynamic. And these are the messenger RNAs with the immediate early genes, delayed early genes. Those are microRNAs. And there is kind of a mirror picture. There are, at time 0, many microRNAs are upregulated to begin with. Once we stimulated, within 20 minutes, many of them come down. They are down-regulated. For example, there are microRNAs that regulate C-FORCE and C-JUN and C-MYC. Turned out that these are indeed microRNAs that regulate the oncogenic transcription factors. And overall, this is an example of C1. Overall, we found that these microRNAs, a physiological negative feedback control, are defective in cancer. This is a complicated and busy slide, but the message is that we looked at several tumors for breast cancer and compared the fold change of tumor versus spared tumors, the ratio. And what we found, here you see the blue, which represents the expression of those microRNAs, suppressor microRNAs, which are commonly downregulated in tumors versus the nearby normal tissue. And these are the microRNAs labeled here. So again, another example of feedback control, which is defective in cancer. And if I integrate now the delayed early genes and the microRNAs that stimulate them, what we come up is this model. Namely, at time 0, there is little microRNA, messenger RNA made for the transcription factors that are oncogenic. In parallel, the microRNAs that regulate them, degrade them, they are up. When we stimulate with EGF, within minutes, the microRNAs go down to allow the transcription factors to come up. But slightly later, the delayed early gene in green, they come up to suppress the oncogenic transcription factors. So this is a complex transcription control mechanism, and as I showed you, it is defective in cancer, as you can see here. So coming back to the issue of complexity and lessons we learned from mathematicians. For example, we learned from mathematicians who build networks in their computers and then attack them in an attempt to destroy them. So what they tell us is that networks are trained through network training to overcome common perturbations. They show extreme fragility to uncommon perturbation. And this is what we would like to find out. What are the uncommon perturbations that will fail such oncogenic networks? Also, robust networks are hub addicted. They develop reliance, extremely reliance, on some hubs. Glucose is one hub. ATP is another hub. PI3 kinase, AKT, mTOR is another hub. So if we intercepted those hubs, we would probably collapse the network. And I would like to give you, very briefly, several examples for uncommon perturbation. The first one is the use of immunotherapy, cetuximab, trastuzumab, that bind to cancer cells like this yellow labeled loaded tumor cells. Here it is, loaded with a fluorescent dye. And once the antibody is there, it recruits T lymphocytes to inject toxic materials, make big holes in the membrane to allow the dye to come out, and overall cell death. ADCC, antigen-dependent cellular cytotoxicity. This is very uncommon. It has to do with autoimmune response, but we do not know of any autoantibodies to those receptors. So very uncommon perturbations. Another uncommon perturbation, not yet a drug, experimental, are those chaperone inhibitors like geldanamycin or 17-AAG that bind to HSP90 and in turn cause degradation. For example, HER2 is degraded in the proteasome. This is taken from the work of Neil Rosen and David Solid. HER2 is degraded rapidly after treatment with those drugs. And another uncommon perturbations are those drugs that bind to the ATP binding site of the receptor, like erlotinib and some other drugs that inhibit kinase activity. What we learn is that the drugs that target simultaneously, more than one target, they are relatively effective, like lapatinib in this particular example. And the reason why they are particularly effective is because the network was trained to resist one perturbation at a time. Mutations are very rare in evolution. So if we combine two perturbations at the same time, this is very uncommon and did not appear while the network was trained to resist perturbations. My last example will take us to my own work, in which we combine two antibodies to either HER2 or EGF receptor. What we find is that in some cases, when we combine two monoclonals to HER2, we see complete inhibition of tumor growth. So these are tumors grown in mice, in animals, of HER2 overexpressing cells. They give rise to tumors. But when treated with a single antibody, there is some inhibition. But when treated with combination of two antibodies, very large inhibition. What we found is that when we combine two antibodies, we are able to degrade EGF receptor. In some cases, in this case, for example, this is EGF receptor. EGF causes, of course, endocytosis degradation. But this is a combination that was not so effective. Turned out that they were not effective because they bound to the very same site. They were competitory. They are not going to synergize. So we come to this conclusion. When we combine two antibodies, like the black and the yellow, they make large complexes at the cell surface. They collapse into the cytoplasm. They go to the lysosome, just as you saw in the movie, except slowly, and degrade. And this is a very uncommon perturbation that I hope will be translated to clinical application. So I'm summarizing. This is my last slide. Efficacious cancer therapies may explore fragile aspects of oncogenic networks like uncommon perturbations that the network in the last 800 million years, during its training, the network was not exposed to immunotherapy or combination of two perturbations or combination of antibodies or HSP inhibitors. Also, resistance to drugs reflect system plasticity and may be overcome by drug combination. And this is my major and general lesson. Evolution transformed relatively simple cascade network, linear cascade, into robust, multi-layer, heavily complex networks. And it trained them to overcome single perturbation, that in your language, single perturbation is monotherapy. Evolution did not train those networks to overcome complex, simultaneous perturbation, what you call combination therapy. This is very uncommon. Also, system control is defective in cancer, and I gave you several examples like receptor endocytosis and feedback loops that have to do with messenger RNA and microRNAs, which is a very recent development in the field. Thank you very much for your attention.